And good morning. My name is Kamila Stokowa-Soltys, and I would like to thank the organizing committee for the opportunity to show my results. And today I will say just a few words about cupric complexes with peptides constituting fragments of outer membrane proteins from Fusobacterium nucleatum. At the beginning, I would like to familiarize you with the premises that led me to undertake such research topic. So Fusobacterium nucleatum is one of the most abundant gram-negative anaerobic bacteria, part of the good and oral floral commensal flora generally found in the human dental plaque. However, Fusobacterium nucleatum migrates from its primary site of pollinization to other parts of the body, and it was shown that this bacteria is actively involved in colon cancer progression. And we have shown that Fusobacterium nucleatum outer membrane proteins may take part in cancerogenesis, and therefore it is very interesting to study their interactions with metal ions. And the full characteristic of the coordination process was possible because of the usage of a broad spectrum of experimental methods like UV-Vis, CD, potentiometry, and EPR, and computational ones. So today the coordination part will be treated with neglect, and I will focus on reactive oxygen species generation and plasmid DNA degradation. We have seen some interesting correlations, namely positively charged peptides generate hydroxyl radical in the presence of hydrogen peroxide and even more effectively in the presence of ascorbic acid. Complexes with peptides shown on the slide were also able to produce a superoxide anion radical. However, I didn't attach the spectra. So these complexes, these positively charged peptides, when complexed with metal ions, produce high amount of reactive oxygen species, which are able to cleave plasmid DNA, as I shown here. But we have also seen that free, uncomplexed peptides are able to cleave plasmid DNA. It was shown that the DNA degradation properties do not require redox-equivalent metal ions and/or oxidative conditions. Most probably, the DNA cleavage goes via nucleolytic mechanism, and basic amino acids interact with phosphate group, and therefore acid-base cleavage is observed. On the other hand, complexes with negatively charged peptides produce higher amount of hydroxyl radical in the presence of hydrogen peroxide, but much less hydroxyl radicals are produced in the presence of ascorbic acid. Even free copper-2 ions produce more hydroxyl radical in the presence of ascorbate. So complexes seems to have antioxidant properties. So the question was why it happens. And most probably, negatively charged carboxylic group repel ascorbate, and this molecule cannot reduce copper-2 ions. As we can see on the DFT model, copper ions, colored with the yellow, are surrounded by the peptide and are not solvent exposed. Moreover, carboxylic groups in the axial position of the metal ion, so negatively charged ascorbate is repelled. In consequence, less linear form of DNA is observed, and DNA degradation process is less effective. So summarizing, different peptides bind metal ions with different affinity, and such formed complexes might cause DNA degradation in the presence of hydrogen peroxide and ascorbic acid, and the form cause reactive oxygen species production. Of course, these reactive oxygen species may destroy nucleic acids, may destroy also proteins, carbohydrates, but the damages of nucleic acids is particularly very dangerous, and they may be involved in cancer progression. At the end, I would like to thank Professor Robert Wieczorek for DFT calculations and Alicja Misiaczek for her assistance in the research.